Let me welcome you to the next session of Biochemistry and Applied Zoology, session 17. And in this class, we are going to discuss about enzymes. At least certain basic things of enzymes. And this is of Unit 2. And in that second chapter, the chapter includes classification, properties, mechanism of enzyme action, including induced fit theory, factors affecting enzyme action, coenzymes and inhibitors, as well as biological importance of enzymes. Out of these, in today's class, we will see classification and properties of enzymes. The objectives of today's class are to know the meaning of enzymes, explain the types of enzymes, list out the properties of enzymes. Type here refers to the classification. Outcomes are to be enabled to classify the different types of enzymes, describing the different properties of enzymes. Now, what is an enzyme? This enzyme is a biocatalyst. So, now question is, what is a biocatalyst? To understand what is biocatalyst, you must know what is catalyst. A catalyst, as you have studied in chemistry, is any substance that increases rate of any chemical reaction. Now, what is rate of chemical reaction? Rate of chemical reaction is very simple. It is how many of the substrates are converted into products within a given time. Let's say that if 1000 substrates are converted into 1000 products in 1 minute. This is one case. 10,000 substrates are converted into products in 1 minute. This is another rate. So, rate of chemical reaction is nothing but it is the number of substrates getting converted into products, and this number of conversion within a given time varies from one reaction to another reaction. And you all know that there are certain reactions which need additional external energy. And sometimes some of the chemical reactions will release energy by themselves. Similarly, our body is formed of cells, and cells perform all these biochemical reactions. Our cell is a small laboratory in a way. And number of biochemical reactions take place in this particular cell. That means there are certain chemical reactions like your chemistry lab, and similarly there are also reaction rates. That means every chemical reaction has some particular velocity, speed at which the conversion takes place. And some of the chemical reactions their rates can be increased, enhanced by using certain additional chemical compounds or molecules. Chemical compounds in case of chemistry in a normal chemical reaction that takes place in lab, whereas molecules in case of the lab that is present inside our body, that is the cell. So that means a catalyst, a biocatalyst is that substance, truly speaking, that molecule which will increase the rate of biochemical reaction, but without undergoing any change itself. When I am saying any change, it refers to chemical change. So, now in case of chemistry, you study that the catalyst some of them can increase or decrease the rate of chemical reaction. But we have biocatalysts that will increase the rate of chemical reaction, because in the absence of those chemical reaction, at least if a chemical reaction takes place in a fraction of a second, the same reaction might be completed in 30 hours, 40 hours, like that. So it increases the rate of a biochemical reaction. Why we are using the word biochemical? Because this is a chemical reaction taking place inside the cell, inside a living system. That's why it's called as biochemical reaction. So I'll repeat. Enzymes are biocatalysts. Biocatalysts are those molecules which increase the rate of chemical reaction, biochemical reaction, without itself undergoing any change in the overall process. Okay, so enzymes are biocatalysts, and these are actually biological polymers, and they are most of them, 99% of these enzymes are basically proteins. They are basically proteins. And the concept of enzymes was first started by a French chemist, okay, Anselm Payon, you can say, discovered this enzyme in 1833. In 1836, Berzelius, okay, he coined the term catalysis, okay, that means to dissolve. And in 1878, Kuhn used the word enzyme, where enzyme in Greek means in yeast, enzyma, in, zyma means yeast. Okay, so in yeast for the first time. The name enzyme is literally means in yeast. Okay, and then it was actually the Buckner in 1883, for the first time he isolated the enzyme from cell-free extract. Okay, and in 1926, it was James Sumner first achieved the isolation and crystallization of the enzyme, and the first such enzyme that was isolated and crystallized was urease, and this was done from jack beans. Okay, and then its identification was done, and we know now that these enzymes are basically proteins. Now let's define enzymes. So enzymes can be defined as organic catalyst that accelerates, increases the biological functions of the cell. Okay, so whatever I said in general is put in words here. So most of these enzymes are proteins. They are formed of amino acid sequences in primary, secondary, tertiary, or even in quaternary structures. They will not, whenever, that is a property of the catalyst actually. Any catalyst, when it is involved in any chemical reaction, it might undergo physical changes, but it will not undergo any permanent chemical changes. Okay, and these enzymes, they are highly specific in a reaction. Okay, just like a specific key can open a lock, there might be duplicate keys that we can prepare, but for enzyme and substrate reaction, always they are highly specific. And not only that, they are reversible, in the sense they can be used again and again. Any particular enzyme can be used. Okay, for example, amylase, it can convert thousands of molecules. So conversion is possible because of their reversible properties. Okay, and enzymes, where they are synthesized. These enzymes are, we said that they are proteins, most of them are proteins. So proteins are synthesized by ribosomes, and the information needed for the synthesis of these proteins are present in DNA, which will be then transcribed by mRNA and then translated in cytoplasm by using ribosomes and tRNA with the help of rRNA. And their activities can be regulated, or they can be induced, or they can be repressed. That means they can be controlled. There are feedback mechanisms. If you are going to the structure of amino acids, so enzymes, these enzymes, I said proteins, so they are formed of amino acid sequences, which are arranged, amino acid sequences, which are either in their primary structure, or they might be present in their secondary structures, or they might be present in their tertiary or quaternary structures. Okay. Each enzyme, irrespective of its condition, will have something called as active site. Okay, I said enzymes are the catalyst, biocatalyst, which convert a substrate into a product. So how it converts it? Most of the times a bond is broken or a new bond is formed. The biochemical reaction is a matter of breaking or synthesizing something. It is either anabolism or it is a catabolism. Okay, we are talking in physiological terms. And in terms of biochemistry, it is breaking down or synthesis. Okay, so the enzymes help, they themselves will not be doing something, they actually help in adding a bond or they'll help in breaking a bond. Now, this is done by providing a special space for the reaction. Enzymes provide special space for the reaction to take place. So, such spaces which are present on the surface of enzymes are called as active sites. They are called as active site. So, these are the sites to which the substrates bind. And these sites, I said, enzymes are specific, and these sites are specific because of because of the tertiary structure of the protein. Okay, and this site generally occupies less than 5% of the enzyme surface. They are present on the surfaces anyway. So if by any reason these active sites, okay, are altered, then enzyme activity will not take place. So let's say that this is one particular enzyme, and there might be a small region, okay, very small region. I'm drawing a little bigger one. So this is the area to which a substrate can bind, where the reaction is going to take place. That site to which substrate binds is called as active site. Okay, just like you have a lock and key. Okay, even the lock is much bigger, but the keyhole will be smaller, isn't it? In the same way, active site will be a small portion of the enzyme to which the substrates bind and where the chemical reaction is carried out. Now, this active site will have two regions. One is the binding site, the other one is the catalytic site. Now, we have a three-dimensional picture. Let me go for a two-dimensional one. So, let's say that this is the active site of the enzyme. Let's say that this is the enzyme, okay, and this is the active site. Now, here in this active site, let's say that this is the region to which the substrate is going to bind. Okay, so this is called as binding site. Let me use a different color. So this is the binding site, and there might be a special region here, okay, here in the active site that actually help in breaking this bond. It might be bringing the breaking of the bond. Then, so the active site, this will be called as a binding site, this will be called as a catalytic site, breaking the bond. It might be adding the bond also. Okay, so active site has how many regions? Two regions. One is the substrate binding area and the other one is the catalytic. But catalytic site will be always very close to the binding site, because if it is far away, a bond cannot be broken or cannot be added. So it will be close to the binding site within the active site. Okay. Now let's see the different properties of enzymes one by one. There will be repetitions anyway. So first property is its catalytic property, that it speeds up, it increases the rate of biochemical reaction. So for conversion of for breaking down of one maltose into glucose and glucose in a normal reaction, if it takes 24 hours, in the presence of enzyme maltase, it will be converted in fraction of a millisecond. Okay, so it speeds up the chemical reaction. You imagine one conversion, if it takes 24 hours, how many how long it may take to just to break 1 gram of maltose. Okay, whereas in the presence of maltase enzyme, it will be converted very fast within a fraction of seconds. Okay, and this will not be affecting the nature or property of the end product. End product will, this will, its function is only to help in breaking or adding the bond. This will not contaminate the product. There is a story of uh, white horse. It is like you just add them, and then as soon as the calculation is done, you remove it. There is no change in the actual number of the horses that were taken in the beginning. So it is like that. Okay, so they won't interfere with the property of the end product. These are highly specific. That's very important one. That makes them special. Okay, these are highly specific in their function, and they are always larger than the substrates because they have to provide the active site. These are highly sensitive to the pH because they are proteins. Because they are proteins, they are sensitive to the pH. They are sensitive to the temperature and the substrate concentration. We will be discussing this particular sentence in whole session in one of the sessions. So they are sensitive. You know that the enzyme that is active in your mouth will not be active in your stomach because stomach pH is highly acidic. The enzyme which is active in alkaline pH will not be active in acidic pH. One which is active in acidic pH will not be active in alkaline pH. And similarly temperature also. Okay, and they are strictly stereoscopic in nature, and most of them are proteins with the exception of riboenzymes. In RNAs we have come across this. Riboenzymes are non-proteinaceous enzymes. Okay, yes. Next, this is the picture of catalytic activity of the enzyme. So there is an enzyme, there are two substrates, substrate 1 and 2. Okay, both of these will bind here. I said there is active site, so they bind to the binding site. That which is shown here in somewhat yellowish color is the binding site. So they bind to the binding site, and then these two substrates will be, the bond is added between them. Bond is added between them. Let's say that there is one glucose molecule. Sorry for that. Okay. There is one glucose molecule and then there is a second one. So these two are added. What happens? Condensation of a molecule of water takes place, adding a bond. And the product formed is nothing but, if there are two glucose, it is a maltose. Once the product is formed, it is released. Will there be any change in the, is it going to affect the end product? No, it won't. Okay, this will remain unaltered. It is now ready to receive one more substrate so that it can convert them into product. Okay. Now moving on to the classification of nomenclature and classification of enzymes. Enzymes are, nomenclature means naming, you know. So enzymes are named. In olden days they were given different names by their discoverers, and they were arbitrary in nature. For example, pepsin, trypsin, chymotrypsin and all. Just by saying the names, you cannot identify their function. Okay, so only to differentiate them from the product, they were named, I mean the suffix ACE was used, ACE, ASE was used, okay, in olden days. But they wanted to make it highly specific. We are talking about science, so they wanted to make it so very scientific one. These olden names were called as trivial names. They were trivial. They were not a highly significant one. And then it was International Union of Biochemistry, also called as IUB. It appointed a commission in 1961. The name of the commission was Enzyme Commission. Okay, and this work of this commission was to prepare a scientific way of naming enzymes. Okay, and this commission, after their thorough study, they have formulated some basic principles for classification of enzymes. Okay, and they are naming. And this was completed by 1964. So since 1964, the IUB system of enzyme classification has been adopted worldwide. IUB, International Union of Biochemistry. And now at present we have 6 major classes of enzymes. That too, they should be named in order: class 1, class 2, class 3, class 4, class 5 and class 6. And each one of them have been classified based on their function, based on what kind of chemical reaction they catalyze. Okay, based on that the names are given, the classes are made. Okay, so how many classes are there? There are 6 classes of enzymes. Let's see one by one. The first class is class 1, oxidoreductase. Oxidoreductase, as the name itself says, oxidation and reduction. In chemistry you come across redox reactions, reduction oxidation reactions. So here the name is given as oxidoreductases. See, oxidation reduction is the reaction carried out, catalyzed by them. So these class of enzymes help in increasing the rate of chemical reaction by either oxidizing or by reducing. Either they are going to act as oxidizing agent or by reducing agent. Okay, so they help in oxidation or in reduction. Such group of enzymes are called as oxidoreductases. Okay, and the example class of enzymes that come under oxidoreductases are dehydrogenases. Dehydrogenases, alcohol dehydrogenase for example. Okay, that is one example. Then there is cytochrome oxidase. Okay, L and D amino oxidases. All the enzymes which have a suffix of oxidases, they come under oxidoreductases. Enzymes with the dehydrogenases names come under oxidoreductases. So usually they catalyze oxidation reduction reactions. Let's say that there is a AH2 molecule, okay, and there is a B. The reaction between this will result in A plus BH2. Addition of hydrogen is reduction. So this is reduced. This is oxidized. So any enzyme involved in this reaction is called as oxidoreductase. It is a oxidoreductase. Next one is a transferase. Class 2 is a transferase. As the name itself says, they are involved in group transfer reactions. They transfer functional groups from one substrate to another. Group transfers. Functional groups you are familiar. Aldehyde functional group, keto functional group, then there might be carboxylic acid functional groups. So this kind of functional groups are transferred from even phosphoric acid. Phosphates are transferred from one group to one substrate to another substrate. Okay, so if any enzyme is involved in transferring the groups, functional groups, then they come under the class transferase. Okay, so examples for these are hexokinases. These transfer phosphates from one substrate to another substrate. Then there are transaminases. Amino groups are transferred. Then there are transmethylases. Then phosphorylases. Phosphorylases and hexokinases both are involved in transferring the phosphates. The name hexokinase is given because this is a enzyme that will transfer the phosphate group to the hexose sugar, that is you can take glucose or fructose. Okay, so hexokinases are examples for transferases. They are involved in transferring a group. Let's say A. Let me use a brighter color. So X. This is X is functional group here. Okay, and then we have the substrate, second substrate. Now this X functional group is transferred here. So what happens? A plus BX. This X might be phosphate. Okay, it might be phosphate. Then the enzyme is phosphorylase, and if A is sugar, then it becomes this B is a sugar, it becomes a hexokinase. The third class is class hydrolase. As the name itself says, hydrolase, hydrolysis reaction. Hydrolysis is a reaction where a molecule of water is broken down and added across the bond. Such reactions are called as hydrolysis reactions. So here what happens is, so let's say that there is AB, this is 1, okay. Now a water molecule is added, H2O. Now what happens during the reaction is this H2O is broken into H plus and OH minus. Hydrolysis is taking place. And then this H plus, okay, it is added to A. So AH plus this is added to this. So BOH. Now what has happened? This bond is broken. That bond is broken. Okay. So any chemical reaction where a molecule of water is broken down and added across the bond, resulting in separation of the substrate into two different products, then you call such reaction as hydrolysis. Any enzyme involved in such reaction is hydrolytic enzyme belonging to class hydrolases. All are digestive enzymes like pepsin, renin, okay, amylases, they come under hydrolases, hydrolytic enzymes. Lipases, esterases, okay, phosphatases, pepsin, urease, all digestive enzymes come under this. Next class is class lyases, lya, breaking down. So these are again involved in breaking a bond, particularly, okay, they are actually involved in breaking a bond, lyase, okay, the meaning of lyase is breaking. Now usually what happens when the bond is broken, most of the times a double bond is created, okay, and this double bond is created generally by adding or removing a substance, generally carbon. Now this is different from hydrolytic reaction because here there is no breakage of water. Hydrolysis reaction is characterized by breaking a water molecule into H plus and OH minus. But here there is no breaking of water molecule into H plus or OH minus. Any other reaction other than hydrolysis where a double bond is created or a bond is broken can be considered under lyases. Aldolases are the best examples for lyases which you have studied in glycolysis. In glycolysis you come across this particular enzyme called aldolase, and this is a classic example for lyases. Okay. In addition to that, there are also fumarases and histidases. Okay. The next one is isomerase. Very simple. Isomerization reactions are catalyzed by these group of enzymes. Enzymes involved in isomerization. What is the isomerization reaction? It's a reaction of converting one isomeric form to another isomeric form. Okay, any enzyme that converts one isomeric form to another isomeric form is called as isomerization reaction. Enzyme involved is called as isomerase reaction. Glucose 6-phosphate is converted into fructose 6-phosphate. Glucose and fructose are isomers. Glucose and fructose are isomers, and here the conversion from one isomeric form to another isomeric form takes place by the help of enzyme isomerase. Okay. Next one, example of what you see on the screen is a phosphoglucomutase. Then there are also triose phosphate isomerase, okay, retinol isomerase, then phosphohexoses isomerase, phosphohexose isomerase. That is the enzyme which will convert glucose 6-phosphate into fructose 6-phosphate. Okay. The last class of enzyme is class ligase, and this forms new bond, ligation, joining. It joins the bonds. Okay, and classic example for it is DNA ligase. So they are involved in synthesizing some substances. So they are actively participating in synthetic reactions. and usually ATP molecule will be used. So DNA ligases, they join the strands of DNA. Nucleotides are joined by the help of this kind of ligases. There are also all different classes of synthetases like glutamine synthetase, okay, acetyl coenzyme, carboxylase, succinate thiokinase, etc. They are called as ligases. Okay. Condensation reactions are where ATP dependent condensation reactions are catalyzed by these kind of ligases. So there are 6 classes of enzymes. Class 1 oxidoreductases that carry out oxidation reduction reaction. Transferases, they help in group transfers. Hydrolases break bond by adding a molecule of water. Lyases break bond other than hydrolytic reaction. Isomerases convert one isomeric form to another isomeric form. Ligases join different molecules. Ligation joining. So that's all for today's session. Summary of the class are: enzymes are biological catalysts. Most of them are proteins. Okay, they possess active sites and are highly specific and are sensitive to pH and temperature. Based on their function, they are classified into 6 classes. The word enzyme was coined by Anselm Payen, Wilhelm Kuhn, Louis Pasteur, Robert Koch. So it was Kuhn who named enzyme. Enzyma means in yeast. The first discovered enzyme is maltase, polymerase, diastase, isomerase. It was diastase, which is not a property of enzyme. Non-specific, sensitive to temperature, to pH, to substrate concentration. Yes, the answer is they are non-specific. They are not non-specific, so non-specific is not the correct answer. The substrate binds to dash region of the enzyme. Prosthetic, apoenzyme, cofactor, active site. Answer is active site. Enzymes which catalyze the joining of specified molecules or groups by double bond. Joining, okay, of molecules. Transferase, oxidoreductases, lyases, ligases. It is the lyases. They create double bonds. Thank you.